Hello everyone, my name is Binay. Welcome to my channel LifeLore. Today we are going to discuss about different types of blood cells present in your blood. If you are seeing my video first time, please subscribe to my channel and also like and share my videos so that you can see more videos like this. So here today we are going to discuss about different types of blood cells present in our blood. So first, blood, it's a connective tissue. So blood usually in an adult, it presents around 5 liter and it consists of around 7% of our total body weight. So what are the components of blood? There are main two components. First is plasma and another is blood cells. So don't confuse plasma with the serum. Plasma is the liquid part of the blood and the serum is different. I'll talk about that later. So plasma, what is plasma? Plasma is, suppose this is our blood vessel and these are the endothelial cells. So blood vessel lining is called endothelial cells. And the liquid part, apart from the blood cells, all the liquid part in our blood is called plasma. And it also consists of different clotting factors. So that is the difference between plasma and serum. What is serum? So serum, when we keep our blood for some time, it will become clot. So clot, which contains also different fibrinogen and also these cells. When we remove this, the other from blood, the other part will be serum. So the serum has no clotting factor, but plasma has everything except blood cells. So that is the basic difference. Now coming to the blood cells. So what are the main blood cells present in our blood? So there are two types, basic two types. First one, red blood cell, RBC, and white blood cells, WBC. So red blood cell is the main part. It constitutes around 40 to 50%, around 45%. 40 to 45%. So this is the main part of our blood. And WBC, total WBC is around 1%. WBC is 1%. Other component is totally plasma. So this is the main constitution of our blood. So RBC is red blood cell and also called as erythrocytes. We'll talk about that later because we have in another video we are going to discuss about erythrocyte in detail. So please subscribe to channel to see that. And so just for information, if we reduce, suppose we constitute 40 to 45% of our blood. So if this is reducing, so this is called anemia. Because erythrocyte carries oxygen to our tissue. So reduction of erythrocyte will reduce the oxygen level in our blood and it will not, the cells in our tissue will not function properly with lack of oxygen. So this is RBC, and apart from RBC there is another cell called megakaryocyte. Megakaryocyte produces platelets, which are important for our blood clot. So this is also another type of cell which is also produced from common erythrocyte progenitors. And apart from this, we have now WBC. WBC has three parts: granulocytes, monocytes, and leukocytes. So why granulocytes? Because these cells have granules in their cytoplasm. That doesn't mean that all other cells do not have granulocytes. That means these cells have specific kind of granulocytes. That is why they are called granulocytes. The first kind is mast cell. This is mast cell. So this is a kind of cell which produces different cytokines with response to different inflammations. So usually these kind of mast cells are present in our skin tissue. So another is basophil. These granulocytes are stained with basic dyes. So that is why these are called as basophils. Basophils are non-phagocytic cells, and they secrete histamines in response to inflammation. Basophils, they consider basophil and mast cell are very much similar kind of cells. These basophils are present in blood, but these mast cells are present in tissues. Basophils are present in our blood around less than 1%. So usually when we have parasitic infection, these basophil cells will increase. So that is the when doctors check these basophils are increased, first thing they consider is infection with parasites. Next is eosinophil. These kind of cells, and these all these granulocytes are multi-lobed. Actually, these two kind of cells, basophils and eosinophils, are bilobed. This is the nucleus looks like two lobes, so that is called bilobes. So eosinophils stain with acidic dye. And these are also formed with response to inflammation, usually to allergic inflammatory response. And these are somewhat phagocytic cells, and they destroy larger parasites and microorganisms, and they constitute around 1 to 3%. If this is increased, you will see hyperreaction in our body, hyperallergic reactions. Next is neutrophils. Neutrophils are the most common kind of cells present in our body, and they constitute around 50 out of all the WBC. They constitute around 50 to 70% of WBC, and they are the primary response cells in response to our inflammations, and they are multi-lobed. When you see these cells, they have multi-lobed nucleus, and they are neutrophilic they are neutrophilic to the dyes, and these are the most phagocytic cells with response to different inflammation. They respond first, and they phagocytose the microorganisms. So next is monocyte. Monocytes are two kinds, dendritic cells and macrophage. These two monocytes constitute around 1 to 6%. Dendritic cells are the antigen presenting cells. These two are antigen presenting cells. Dendritic cells are less phagocytic than macrophages. When we see inflammation, the macrophages phagocytose those antigens and present to phagocyte. After phagocytosis, they present dendritic cell and macrophage. They present these cells, these antigens to B cell or T cell, and they help in the differentiation of B cell and T cells. So they are present in periphery of our blood. So this is dendritic cell, and this is macrophage. And macrophage contains different kind of lysosome enzymes, and they help the macrophage in the destruction of antigens and processing. So next is leukocyte. Leukocytes are three types. First is B cell, T cell, and natural killer cells, NK cell. B cells are antibody producing cells. T cells can be helper T cells or cytotoxic T cells. So usually in response to the antigen presenting cell, they can be helper T cell or cytotoxic T cells. So these T cells helper T cells can activate other B cells and other inflammatory response, and the cytotoxic T cells usually kills those infected cells, and these leukocytes are produced mainly because of viral infections and some kind of chronic inflammations, also chronic infections. And T cells, these cytotoxic T cells can kill those infected cells, and natural killer cells are also similar to T cells. These natural killer cells, they kill these infected cells. Usually viral infected cells are killed by natural killer. These lymphocytes are usually, these T cells are mature in lymph nodes, mainly thymus. So that is why these cells are called lymphocytes, and these cells constitute around 20 to 40% of our WBC. WBC is otherwise called as leukocytes, and this don't confuse with lymphocyte and leukocyte. Lymphocytes are one of the leukocytes. So how do you differentiate these cells? These cells can be differentiated under microscope. So as I said, based on different dyes, the specificity towards the dyes, these cells are recognized. So usually what we use for the recognition is HE stain. So H is hematoxylin eosin stain. When you put some one drop of blood on the slide, so you put the blood here, then you take another slide, and then you make a smear. Now you see the smear, and then keep some time for dry. Then you stain them with HE stain, and you see under the microscope, you'll see different kinds of cells. So you can recognize these cells seeing those morphology. So you can see clearly the megakaryocytes will be bigger cells, and RBC will be the smaller cells. You can differentiate clearly these two types of cells, because RBC will be the main part of those cells, constitution of those cells. And mast cells, as I said, they have nucleus, single nucleus, and basophils, they stain with basic dyes, and they will look the granules will look blue, and eosinophils will look purple or red or pink. And these two kind of cells will have bilobed, and you can see clearly the cells which have multilobed nucleus, those are natural killer cells, and these cells will be the maximum after RBC. And these dendritic cells, you will see the different protrudings, and you can differentiate these cells. And macrophage will also look bigger. And these cells, these lymphocytes, will have the bigger nucleus. They have biggest nucleus, and they have very less cytoplasm. These but you cannot differentiate between B cell, T cell, natural killer cell by looking those under microscope. So for that differentiation you need flow cytometry. We will discuss that later. So this is all about different kinds of cells present in our blood system. So thank you for watching my video. Bye bye.